easier. Okay. And let me read the invocation real quick. Dear Lord, we thank you for bringing us together as friends and Rotarians. Thank you for this beautiful planet. Thank you for the fellowship of Rotary around the world. Help us to build a world based on fairness and goodwill and truth. Give us peace in our time. Bless all the people of the world. And in the name of God Most High, we pray. Amen.
Amen.
Now, our four-way test. And also, this is starting tomorrow, is World Immunization Week. And obviously, we aren't out, people aren't out giving polio vaccinations right now, going door to door or going in communities and all. But when we're back together, or when people can get together and we can do this again, we certainly want to start it again. But you can go to the Rotary website and find out more about Rotary World Immunization Week. And now, remember I told you we want to do, we want to have good news. And I'm going to start. This is a picture. This is my good news. This is a picture of one of my nieces. Her name's CADY. And we were all pretty scared until like yesterday, because CADY had the virus, got the virus. She works in publicity in Nashville, Tennessee, and she got the virus. And a friend, she thinks she got it from a friend of hers, and because she was in lockdown and everything, and came down with the virus while she was locked down. And so just yesterday, she got better. She got better once. She was really sick, just sleeping all the time and just felt miserable and everything. And then she started feeling better. And then all of a sudden it came back and she was like back at square one and felt horrible again. But as of yesterday, she's feeling a lot better now and doing well. So that's my good news. And Diane, I think you have some good news to share.
I do, Marcia. First, Don Parks, Bill Goodwin, and Chris Gaynor all say hello. They're all doing well. The other news I have is one of our favorite groups that we sponsor every year, the Emerson Speech and Debate Club from the school, is still competing online. And out of 200 plus entries last week, they had 5 people. So now they'll go to state and state, but initial competition 2 weeks. So I thought that was great news.
They're all long.
Oh, it's wonderful, Diane.
Diane, hi, this is Philip.
Yeah.
Can you forward me the contact information for I think it's Jeremy. He's the club sponsor.
Hold on.
You don't have to do it now. Just sometime in the next couple days.
Sure. Yeah, absolutely. I'll forward the email to you.
Great, thank you.
Sure.
Okay, and does anybody else have any good news they'd like to share? Anyone? I heard a voice start to speak there.
Yeah, we have mixed news. Our son Trevor and his wife have tested positive for antibodies, but they never tested positive for the virus.
Wow. Wow. Well, that's good. Maybe there's just something in their bloodstream that.
Well, we don't really know what it means. They might have. It's possible that they would have tested positive earlier if they'd been tested, but we don't know that for sure.
But doesn't that mean having the antibodies is they've had the COVID-19 and it's gone past it? I mean,
well, assuming the antibody test is accurate, it's possible. Yeah. It's possible it's a false positive.
Yeah, this is Dr. Bronstein, Jeff Bronstein.
Dr. Bronstein.
Hi. So yeah, depends on the antibody test, but in general, what it means is you've been exposed to the virus in the past, whether you were symptomatic or asymptomatic. But as you mentioned, the accuracy of the test depends on where it where it's coming from. The tests that are recommended by the FDA that have been approved, the ones that have been used in the California studies, have about a it was about a 1.5 to 2% false positivity rate. So, so we'll have to see what it means. Even if you have it, we don't know if that makes you immune or not. Although if you've had it before, your immune system has a memory. So it's likely that if you get it back again, it's not going to be as bad. So still a lot to learn and what these tests are. That's why I don't recommend running out to get antibody tests and those sorts of things right away until we know more about what they mean.
Well,
Thank you.
Thank you, Dr. Bronstein. And I'm going to turn the meeting over right now to Carol. And she is going to introduce Dr. Bronstein, and I'm going to take my screen share off so he can put his up, okay? So Dr. Bronstein, can you, there you go, okay.
There it is.
I'm going to put myself on mute now.
Am I on? Can you hear me?
Yes.
I see you.
May I introduce Dr. Bronstein? Are you there, Dr. Bronstein? I'm not hearing him.
Dr. Bronstein is on, and he's got his PowerPoint up.
And he's
Okay.
muted.
Can you?
Yeah, I was muted. It switched.
Okay.
Okay, yeah.
So can you hear me, Dr. Bronstein? It's
I
Carol
can.
Rosenstein.
Hi,
Hi,
Carol.
good
Great.
morning. This is so amazing to be meeting you like this. I feel so honored for all of our club today. Dr. Bronstein, of course, is no stranger to you all. Dr. Bronstein is an illustrious member of the scientific community. His patients adore him. And Dr. Bronstein, we have 7 patients here today, eager to hear your sharing of all of the latest news in Parkinson research. We also have a colleague of yours, Dr. Ross Goldberg, who's a well-known radiologist in town, and our board chair who wants to say hello for 1 minute. Can you hear us, Ross?
I can, thank you. And I just wanted to say how fortunate I am to have met Carol many years ago, 6, 7 years ago, got me involved in this charity. And I thank her for the opportunity. And I am always in awe of her energy and commitment and what she does to make this all work for Music Men's Minds. And I also, Dr. Bronstein, have heard a lot about you over the years being in the community. So when Carol told me you would be speaking, I couldn't resist the opportunity to hear what you had to say about things, since you're one of the world's experts in Parkinson's and dementia. So thank you.
Thank you.
Thank you. Thank you. Thank you, Dr. Goldberg. So to further introduce Dr. Bronstein to you, we have a very personal relationship with Dr. Bronstein. He's been our personal neurologist for many years. It was Dr. Bronstein who changed Irwin's medications when he was hallucinating and said, we got to bring the dose down. And as I watched Irwin playing the piano and saw him reconnect with his environment, I ran to the phone and said, Jeff, I'm looking and seeing Irwin re-engage in spite of his having low-dose med. And Dr. Bronstein said, you're watching the power of music changing brain chemistry. And on that alone, I thought, wow, I've got to fix it for a lot of people struggling with neurodegenerative decline. So we started Music Men's Minds. And of course, here we are together with Rotary International clubs helping take Music Men's Minds globally to over 200 countries. I am on bended knee to say thank you to Dr. Bronstein for being so important in my life personally, and how magical he is for all of us to be able to find some wellness along the way. And thank you, Dr. Bronstein, for just being in our life and just a hero for the Rosensteins and for the nonprofit and for humanity at large. God bless you for all your work. Thank you.
Well, thank you so much for all the kind words. It's
I don't hear anything. Nothing.
Hold on, I see if I was muted. Okay, can you hear me now?
Yes.
Yes.
Okay. Yeah, it says screen changes a little bit when I'm here. If everybody could mute their phone, their video, so that way we won't get a lot of background sound. And then at the end, when you have questions, if you use the chat button on the bottom, that usually works really well. And you can ask questions if you just say chat with everybody. And then you can have questions. That usually works out really well.
Because there's good
So,
stuff.
oh, what happened here? All right. Let's get started. Anyway, thank you so much, Carol, and thank you so much for everything you've done for Music Men's Minds. It's amazing. I could talk forever on how great it is. But as usual, I have more to say than I have time to say it. So I'm going to try to go through some of this stuff pretty quickly so that I have time at the end to take some questions because it's always forget to say certain things. So I think most people know that Parkinson's is a big problem in our country. There's about 1 million people with it. It's a disorder that's more common when you get older. Though the peak is around 62 years of age. Men get it a little bit more than women for reasons that we don't know. And it's probably getting a little bit more common with time. And maybe some of the things I'll talk about can explain some of that. I really want to focus a lot on the new therapies that are being developed. But for me to be able to talk to you about that, I need to kind of give you a little bit of a background about the disease process and the problems. And then when you'll understand why people are trying to develop these new medicines in the direction that the way they're trying to do it, because there's some interesting strategies, and you need to know the background for that. So the first question is, what causes it? And the simple question is, there's probably no one cause. It's almost certain that it's a constellation of causes. And I'll talk a little bit more about that. But like almost all diseases, it's a combination of your genetic background and environmental toxins and things that are in the environment. We know some of these. This is what I spend my life studying, are the environmental causes and some of the genetic causes. So I know a lot about it if people have questions on that. And I'll talk very briefly about it during this talk. But it's this interaction between genes and the environment. Now, when we look in somebody's brain with Parkinson's, the universal, I don't know if you can see the pointer, the universal feature, the defining feature in the brain of somebody with Parkinson's is this thing where the arrow is pointing, which is called a Lewy body. That's abnormal. The brown stuff around it is actually pigment, which is normal, and that tells us it's a dopamine producing neuron. So it's this accumulation of protein in the dopamine neurons that was discovered many years ago by a Dr. Lewy. And if you don't have those in your brain, it's not Parkinson's. So that is the defining factor. Now, it turns out through many studies that what that Lewy body is filled with is this protein called alpha-synuclein. And here in this cartoon, I just trying to show that normally alpha-synuclein is a simple protein that lives by itself. but it can form clumps. In this slide, they're called oligomers. And those oligomers are very soluble. Some of them are very toxic. And it's those toxic oligomers that we think are what causes the toxicity to those dopamine neurons and actually many other neurons. They also can form what are called fibrils, which are very hard to get rid of. And that's what the Lewy bodies are actually filled with, is those fibrils. But we think it's these oligomers that are here that are really the toxic part. Now, one very important finding that we've had in the last decade or so is that the pathology actually spreads from certain parts of the brain in the brainstem down here shown in black, and also in the olfactory bulb. Those are the earliest times that we see it in the brain. And then it kind of moves its way up and then eventually progresses, spreads throughout the brain. So it's a very, very typical pathway where we see this pathology with time. So as the disease progresses, we see Lewy bodies in all these different parts of the brain. And it turns out that people have even looked outside the brain, and it turns out that it's in the gut. It's in the gut even years before people have any symptoms. And one very interesting study, or actually two studies now, where if you cut the nerves from the gut, you actually have a much reduced low chance of getting Parkinson's. This was called a vagotomy many years ago. We don't do it anymore. It was for ulcers. But it turns out that people who had ulcers, and they were fine otherwise, they cut that nerve that connects the gut to the brain, and the risk of Parkinson's went way down. So what we think is shown up in the top slide is that this whole process starts in the gut and in the stomach, connects to that with that nerve called the vagus nerve, and it spreads through that nerve into the brain and then throughout the brain, shown up here. And there's this very, very fascinating process called templating, where a normal synuclein can be coerced into forming these oligomers or these abnormal aggregates and self-propagate. This concept won the Nobel Prize by a guy up at San Francisco for a rare disease called prion disease, Creutzfeldt-Jakob disease and mad cow disease is an example. It turns out that it's the same process. It's just in Parkinson's, it's not infectious, number one, and it's much slower. So that what happens with prions and in mad cow happens over a year or two. This happens over several decades in Parkinson's disease. So I really like this slide. It's a little bit busy and I borrowed it. I forget where I borrowed it from, but it really kind of puts all this together. If you start from the left side here, say 20, 30 years ago, 30 years before people have say a little bit of a tremor, or we figure out they have Parkinson's, they have some problems with smell, a little constipation problem, or they can have sleep problems as well. They start talking and screaming in their sleep. That can be the precursor. It's been called prodrome, but it turns out that it's actually the beginning of the disease. So it's these alpha-synuclein clumps, or the Lewy bodies, that form in the gut, and they form in the areas of the brain that affect smell, affect sleep. And then it's only until they spread into the dopamine cells that you get tremor and you say, oh, all of those things that you thought you're just getting a little bit older turned out to be the beginning of Parkinson's disease. And so this is a really important concept, because if you're thinking about therapy, you know, or stopping the disease, you're halfway through it by the time you know you have it. So we have to keep that in mind when we're thinking about this. It also tells you how slow the process is if this is going on for decades before you even know it. So as a researcher who's trying to study the cause of it, we have to think about what happened to you 30, 40 years ago, and not just what happened to you a few years before the symptoms came on. All right. So that's my little background on what's going on to set you up about what I really am passionate about is how do we treat this disease and develop new therapies. So there are a couple different ways that I like to think about it. First is, how can we prevent people from getting the disease? Well, we can't choose our parents, but we can change what we're exposed to, what's in the environment. So I'll talk a little bit about that. So that's called risk factor identification. We try to minimize it. That's what we do for heart disease now. We don't wait till you get a heart attack. We know if you have high blood pressure, if you have high cholesterol, that's a risk factor for heart disease. So we try to intervene before you get that heart attack. And so we want to intervene before you get the Parkinson's disease. Now, once you get it, we're going to need to slow it down or stop it. And that's what we call neuroprotection or disease modification. So that's the next step in how we think about treating Parkinson's and other diseases as well. And then finally, when people have these symptoms, we need to reduce the symptoms. And so when people have the disease, we want to make their quality of life better, and especially until we find a cure for the disease or something that'll keep it from getting worse. So we've been really good at these symptoms overall. I mean, when I started, or I should say in the early 70s, before I started, before we had levodopa, which is still our best treatment, people lived about 6 years after the diagnosis. Now people have essentially a normal lifespan just from the medicines that we have. Now they're not perfect and they don't stop the progression, but we try to keep up with the symptoms. And so I'll go through some of those developments and strategies. The real home run in this field, the holy grail, is really stopping the progression. And we don't have anything yet, but we have some really exciting things in trials. I've been talking about exciting things in trials for 20 years, and we're getting closer, I can tell you. We're not there yet, but one of these times we're going to get it. And so I'll tell you about some of my favorites towards the end. So what starts the process? So this is a list of all the genes that we know of. Actually, there's more, but these are ones that tend to run in families or dominant or recessive. But if you add all of these up, you can only explain about 5% of all Parkinson's disease. So there are many genes that can lead to Parkinson's disease, many mutations, but it's still a tiny, tiny fraction of all the people with Parkinson's disease. Now, the reason why it's important to find these genes is because it gives us clues in what's causing it. So the way we found out that alpha-synuclein was in those Lewy bodies and made up those Lewy bodies was the very first gene that was found was the gene for alpha-synuclein. And it turned out that they had Lewy bodies in that family that had mutations, but then we looked in people without the mutations, they had alpha-synuclein in the Lewy body as well. So it gives us clues, it gives us ideas of where to look when we're looking for the cause of the other 95%. It's also important to know that even if you carry the gene, like the most common gene is this one called LRK2, LRK2, where there's a number of people have it, it's a dominant disease, but even if you have a mutation, you only have about a 25% chance you're going to get the disease. So even in the genetic forms, there's other things that modify your risk. It may be other genes, and it's certainly the environment. So it's important to realize that even when people have a genetic form, it still doesn't mean you're going to get the disease. So I've been studying the environment in Parkinson's with Dr. Ritz, shown on the right, for many, many years. We have a huge study cohort in the Central Valley of California, We call it the PEG study. And we started years ago because pesticides were insinuated as a possible cause. And if you look real closely, I don't know, can you see my pointer? I'm moving it around. I don't know. You can see here, this is a crop duster. And this is what every day, many of these spray the fields in the Central Valley. Right here, that's a school. And there's a house in this area here. So and through a lot of very sophisticated epidemiology, Dr. Ritz and I have come up with a way to estimate how much and which pesticides people have been exposed to over the last 45 or 50 years through a special registry that these farmers have to submit all of this information for a half a century now. So we have all that information, and we know if you live near where this is being sprayed, we know you're getting a huge dose of it. And we can tell you which pesticides cause the highest risk of causing it. And one of them, for example, this one called Xyram, increases your risk threefold if you've been exposed to it a fair amount. So we know pesticides is a big part of it, but we know it's not everything. And so one of the things that we've spent a lot of time looking at is for some other causes. Most of us that live in a city don't get exposed to a lot of pesticides, but we all are exposed to air pollution. And it turns out that air pollution is probably one of the most important factors. Now, unlike pesticides or certain genes where if you get exposed to a huge amount or have a genetic mutation, that really gives you a very high chance of getting the disease. But with air pollution, it actually only increases your risk a little bit. It's just that so many people are exposed to high level of air pollution that risk over huge populations really means a lot. So it's kind of like COVID. You know, you only have, a couple percent chance of getting it and dying, but so many people get it that the numbers of people dying are huge. 40-something thousand now, even though the risk for an individual is small. That's the way it is for pollution too. The increased risk is relatively small, but for example, in LA, we think from our predictions that up to 70% Parkinson's in LA could be caused from air pollution. So this is something that we're actively studying. This is kind of how I think about what really causes Parkinson's. Is each one of these columns here is a person, and each of the different things on the stacks are different risk factors. So Joe here on the left, Joe has a gene mutation, and so that increases risk quite a bit. But it's not enough to get to this threshold, this yellow line, but he needs to be exposed to, say, a little bit of pesticides, or maybe got in a car accident, a head trauma can do it. And that would put him above this line in why he got Parkinson's, where, Bruce over here got a big dose of pesticide and had some of the other risk factors. Now, most people don't get up to that yellow line, but we all have that potential. So this is kind of how I think about it, and why it's so hard to study is that there's all these factors come together and help determine who's going to get it and who's not. So we're coming up with algorithms trying to identify all the risks in people and all the genes in people. And we can now predict risk reasonably well, but still not good enough yet to say you're going to get it and you're not, but we can tell who's at a high risk and who's not. So we're getting there. Now, we're going to switch to treatments now. And as I mentioned at the beginning, all of our therapies right now really are there to treat the symptoms. And none of them affect the disease course. these are the list of the symptomatic treatments that we have. So we have, where's my pointer? There it is. We have treatments that treat the motor symptoms. These increase dopamine, so this helps tremors that people have. This treats stiffness, difficulty walking. So these are the Parkinson's specific type medicines. But people with Parkinson's also have problems, as I showed you, outside that system, in the sleep centers, in the higher brain regions. So we have medicines that try to help with that. For example, depression is in 3/4 of people with Parkinson's, and it usually has to do with serotonin. So we have drugs that increase serotonin. We have medicines to help you sleep because sleep disturbances are a problem. As I mentioned, the gut is affected. People have a lot of constipation, and we have medicines for that. We have people with blood pressure problems. They stand up, the blood pressure drops, and they may faint. So we have treatments for that. So, and then we have treatments for the side effects of our treatment. So people unfortunately have to take a lot of pills with Parkinson's. We try to minimize that, but as you can see, people can get a variety of different problems. It's probably a good time to make sure I emphasize that everybody is different. And I apologize for those who've heard me say this before. I say it all the time. If you've seen one Parkinson patient, you've seen one Parkinson patient. So not everybody gets everything. Everybody has a unique disease. So what we have to do is personalize that treatment for people. Now, these aren't perfect, so that I'll talk in a second about some of the strategies to improve upon the medicines that we have. When people have certain problems, they're called motor fluctuations, which will show that we can't fix with these medicines. We have surgery called deep brain stimulation, which can really help quite a bit. Again, it's not for everybody, but for certain symptoms. And then finally, we still don't have anything to slow or stop the disease, but we're getting real close. So this is a cartoon of the dopamine synapse. So if an electrical current is coming down from the top and goes from one nerve, and then it has to get to another nerve, which is on the bottom, here where my pointer's going. And what happens is the electrical current comes, and it's, and unlike a wire where they're connected physically, the way we propagate that impulse to the next nerve is through chemicals. These are called transmitters. And this space here is really, really tiny. And what happens is we release a chemical, in this case dopamine. It binds to a receptor right over here, and then it propagates the current. So for the problems with the motor symptoms like the tremor and the stiffness, increasing dopamine or the effect of dopamine is our strategy, because people with Parkinson's don't have enough of this. So levodopa, basically, or Sinemet, which is our best medication, works up here and basically helps the neurons make more dopamine using their own machinery in the right place. So this is probably why it's so effective, and it's the best out of all of our medications, but it has some limitations, which I'll get to in a second. We have other drugs, they're called dopamine agonists, like Mirapex and Requip, for example, where they kind of bypass this whole system up here and bind directly to those other neurons that they're connected to. These are called dopamine receptors, and so they bind directly. Now, they have a lot of advantages because they're synthetic and they don't have some of the limitations, but they also have more side effects and don't work as well. So this is one of the ways we customize our treatment, is taking some of the advantages of carbidopa, levodopa for some people, and take the advantages of Mirapex and Requip for other people. Now, this is a gentleman. Let me see if my videos are working. Nope. I don't know why. All right, well, my videos aren't working for some reason. Let's just see. Oh, okay. So this was supposed to show some of the problems. So this gentleman had a lot of tremor. This is without taking his medicine. And so I don't know if you can see me on the screen, but he'd be shaking. And then 15 minutes after taking his medicines, he'd be moving around like Michael J. Fox. You ever see him squirming around? That's called a dyskinesia. And the reason why that happens is that there's levodopa works really well early in the disease, and if you just take a little bit, it fixes the problem. And then eventually, this is showing the blood levels here on the left. As the blood levels go up, if it's in this range, this kind of orangish range, all the symptoms are pretty much gone. And then when the levels get too low, you take another dose and all the symptoms are better. And this really works well early in the disease. But as the disease goes on, this orange area starts narrowing. The therapeutic window starts narrowing. And so when the same person takes the pills, again, they get better. But if it gets too high, then they move too much. And then if it gets too low, they don't move enough. So this is called a motor fluctuation. So as it goes up, people are better, but then they move too much, then they're better again, and then they're slow, and they start cycling. And this cycling becomes shorter and shorter as the disease progresses, so that some people have to take their medicines every hour or so, or they have these extra movements all the time, or they're stiff all the time. So that is a real problem. And the reason why that's a problem is because the levodopa doesn't stay in the blood very long. It's got a very, very short half-life. So what we're trying to do is come up with better ways to make it smoother. See this yellow line? How can we deliver it in a smoother way? And so we try to do that either giving much smaller doses more frequently, or using other medications. So for example, this medication here called COMT inhibitor, Comtan or entacapone, that makes the levodopa stay in the blood longer. Rytary is a delayed release. So let me show you some of these. So some of these so now we're getting into the newer therapies. So these are some of the newer preparations. So let's talk about Rytary. So this is the newest form, or one of the newest forms of levodopa. It's been on the market now for a year or two. But you can see in the bottom picture here, these are blood levels. So somebody takes their pill, goes up, and it reaches that peak in about a half hour, 45 minutes. And then within an hour and a half, already half of the peak blood levels. And then within 4 hours left here, you can see how much longer that lasts. So many people can keep this going for, can reduce the frequency they have to take their pill because it's longer acting. The problem with it is in some people, this is what it looks like in the top. In some people, it's not as absorbed as regularly as other people. So it can be problematic for some, but it's been very helpful for other people. These just have some of the other preparations that we have. Now, this is called Duopa. This bypasses the whole pills by itself. So in this therapy, what we do is put a tube right into the small intestine that's shown here, and you pump in liquid Sinemet all day long. So the people have to have through a small procedure, have this tube placed, and then the tube comes out, just below the belly button, or just above it, I should say. And then you connect this little pump and you wear it around with the shoulder strap, and you pump it through all day long. And you can now see how the blood levels are stable for the whole day. This is 16 hours. So it really works. It's really remarkable how well it stabilizes it. The problem is you have a tube sticking out of your stomach. You have to carry this heavy pump around. And with these tubes, they get clogged, they have a little infection, so they're not perfect as well. But it did teach us that if we can stabilize these blood levels like we did here, it makes a huge difference for patients. So there's a lot of work going on. We've been participating in a number of trials. We have a new one coming up with better ways of delivering this continuous medication. So this one trials right now, we're at the tail end of the trial. Hopefully it'll come out on the market shortly. This is a drug called Apomorphine, not related to the morphine that people overdose on. This is the agonist like Mirapex or Requip, as I showed you before in that synapse. But unlike Mirapex and Requip, this is really potent and works just as well as levodopa. And it can go through the skin. So shown here is a little needle, and you basically pop that in every morning, connect this little pump, and pump it in all day long. And so this has been really quite effective. It has its problems like everything else, but this will be a big help for many people. And again, it smooths things out, just like the Duopa that I showed you in the previous slide. But as I said, levodopa is still what people like the best. They feel the best on it. So this is a company called NeuroDerm, and there's another company called AbbVie that have the similar technology. I just couldn't find a picture from them. We're starting a study with them actually in a couple months when COVID ends, where you can pump the levodopa right through the skin. The pumps are a little bit bigger, and the needle kind of, there's a small needle that goes through it, but this is actually working quite well. So it's in what we call phase 3 trials, and I'll go through the phases in a second. So this is another way to deliver the therapy continuously. Let me see how I'm doing time-wise. I'm going to speed up a little bit because I want to get to a few things and running on time. This is for people that get stuck. So this isn't to prolong it. This is to get it in people's system faster. So this is an inhaled form of levodopa. So if people's medicine stop working and they take their pill and they gotta wait an hour for it to start working, they can take a quick inhale, and this one kicks in about 5 or 10 minutes. So it doesn't last as long, it lasts an hour or so, but it's a good, we call rescue therapy when people are having problems in this period when we call it off period. So this is now on the market, came on the market. And this is a sublingual form of apomorphine, which is we hear about approval any day now. And this is just like the inhaled one in that it kicks in quickly, goes away quickly, but it's a rescue therapy. It's a little film like those breath mint films you put under the tongue. So this should be out very, very shortly. I'm going to breeze through stem cells really quickly because the bottom line is they're not there yet. We did stem cell treatments years ago. They were called fetal cell transplants, and they did help some of the symptoms. But once you put them in there, we have no control over them. And they actually started getting a mind of their own and releasing too much dopamine in some people. So in many of the people, they get those dyskinesias, those extra movements without even taking the medicine. They were called runaway dyskinesias. And the only way to control them was to have operations, deep brain stimulation. So, and also, remember I said the Lewy bodies spread throughout the brain. It turns out they spread into the transplants as well. So the transplants get sick over years as well. So transplanting stem cells is probably not going to be the answer. But some of the new developments do give us some hope. Number one is we can make them from your blood now. We don't even need to take embryonic cells. They can make it from your blood. We can program them and get more samples so that we think we can overcome some of those problems. But when I really think is going to be the benefit of stem cells is what's called brain in a dish. There's a professor over at Cedars-Sinai who's wonderful at this. And basically what we can do is take people's stem cells, he's already starting to do this, make stem cells from an individual and ask, why do you as a person get Parkinson's disease? We don't need to see your genes. We can see what is wrong with your cells and then customize that therapy just for you. Kind of like for an infection, right? We take your bacteria for your infection, put it in a dish, test the different antibiotics and say, which one is the best antibiotic for your infection? We will hopefully be able to take your cells and say, what is the best treatment for you? And this is really getting exciting now. It's starting to see some real promising results. So this is something probably in the next 5 to 10 years it's exploding. It's in its potential. So I don't think it's going to be there for a therapy yet in 5 years, but I think its utility in the near future is really going to come to pass. So I'm very, very excited, especially about this part of stem cells in a dish for personalized medicine. So what I want to spend my last 5 minutes on disease-modifying therapy. So I'm going to rush real quick. These are phases. So this is a sales like vaccines are in safe trials now. This is chloroquine, phase 2 trials, smaller grow in the large phase 3 trials. And then finally, phase 4 is approval and post-marketing. So this whole process can take over a decade. Now, these are some of this antibody one is something I'm very, very excited about, but there are other strategies as well. One is genetic therapies to keep people from making too much synuclein, small molecules to break up those aggregates. These are all in people right now. These are in phase 1 or 2 trials. I'm going to pass through. I don't have time to do that. But I want to tell you something that's exciting because people have heard a lot about it. So Alzheimer's has a lot of similarities to Parkinson's. It's in different parts of the brain and different protein, but their proteins instead of synuclein are called A-beta amyloid and tau. And they accumulate and they have a spread through a pathway just like alpha-synuclein does. So a company called Biogen actually made an antibody in Alzheimer's patients, and in Alzheimer's disease, we actually have PET scans that can tell you how much A-beta, how much of those plaques are in your brain. So this is an Alzheimer's patient that has all this red and yellow shouldn't be there. Those are all that A-beta plaques and tangles. If you give a placebo after 1 year, no difference. It may be a little bit worse. But the highest dose of this antibody, look at this, it's almost all gone. This is extremely exciting. And when they check the memory in those people, the memory stabilized where it got worse in the people that didn't get the drug. So this is very exciting. And so there's been a lot of ups and downs. So they did the huge phase 3 trial. And this was about a year ago, and it was a big letdown. They say they stopped it. It didn't look like it was going to work. And then they finished parts of the study, re-reviewed it, and said, you know what? It did work. So they've actually applied for approval for this drug for Alzheimer's disease. So in fact, it shows real signs that it's working. In Parkinson's disease, we're doing that study now. So that's in a phase. There's 2 or 3 large phase 2 trials that are in the middle of it right now. For reasons that I don't have time to go over, there's a lot of reasons why it may work even better in Parkinson's disease with less side effects. The real challenge that we have, though, is we don't have that PET scan that shows whether it's getting rid of it. So if it doesn't work, we won't know why. We won't know, did it get rid of the synuclein or not? So that's the problem, but we're going to get results in this year from the first trial. So we're very, very excited about that. And I think I'm really keeping my fingers crossed for this one. So there's a number of trials going on. There's a different type. This is like an immunization where you actually make your own antibodies. That's the one in Austria right now. So there's a lot of activity in this field. In fact, the protein Roche, this was Roche, just released some of the early data. Looks very promising. So with that, I'm to answer some questions. So maybe I'm going to stop sharing.
Okay, well, thank you so much. Really appreciate it. And if everybody can stay on, I mean, I understand that some of you have to go. I understand that because it's just about 1:30 now. But if you can stay on, I'm sure you don't have to get off to a restaurant or anything to meet anybody. Right. So if you can stay on, that would be great. And but if you have
I have some questions. You want me to start answering?
I have a question from Carol, and Carol was asking about why music is so important to some of the people, your patients.
So you know, the answer is we don't know for sure. We do know that the brain is very plastic, right? That means it can adapt. So if somebody has a stroke, for example, if you know anybody has a stroke, they get really weak, but they get some function back, right? And it's not that the stroke went away, it's that other parts of the brain can be trained to make up for those functions. And so the same thing can happen in higher thought process. So we think music is a way of helping train some of the parts of the brain to take over some of the functions that are being interfered. That's the same thing with physical exercise. Huge part. I didn't have time. I had some slides putting together showing the effect of exercise. Exercise actually meets all the requirements for slowing the disease down, believe it or not. And that's, again, the same thing that music does, is it helps tap into our natural ability to retrain the brain. So
Doctor,
that's the best I can do for that answer.
Doctor, at the outset, you said something, if I'm not mistaken, about this disease peaking at age 60, at age 62. Is that correct?
So that's the incidence. That's saying that if I took what's the average age that people get it at, it starts at 62. But the risk actually goes up as you get older. So the average person with Parkinson's gets it at age 62. That's all I meant.
Okay.
Okay, thank you. And Mark Rogo has a question. Do you have a graph that shows how Parkinson's affects adults at different ages? But I think you just said that it starts at 62.
Well, so I can talk. So there is difference. I don't have a graph in that sense. About 15 or 20% will get it before the age of 50. We call that young onset Parkinson's. And it's important to know, remember I kept saying disease is different in each person. So young people like Michael J. Fox, he's got a lot more problems with the dyskinesias, as you notice, than if somebody else gets it at the age of 70 or 80. But he has less problems with memory and things like that, where somebody who gets it older will have more problems with, say, memory and balance. So that's where the differences in the age come into play. So there are different problems in different age groups.
Okay. And from Diane Good, is there any test you can take now to see if you will get Parkinson's, and why not just get a vagotomy?
Vagotomy.
Yeah, to reduce the risk early on.
Okay, well, I'll answer the second question first. So vagotomy is a pretty big surgery, and one of the reasons we stopped it is it's got a whole lot of problems. And if our theory is right, it starts in the gut and then spreads. Cutting the vagus nerve, once it's in the brain, it's already there. So it was kind of like stopping people coming from Europe with the virus. The virus was here. So stopping more from coming in really wasn't going to make a difference. So doing a vagotomy would not stop, wouldn't do any good once you have it. And you don't really want to do a vagotomy on 99% of the population to prevent that 1% from getting it. So, but we are trying to do drugs and things like that. If we can find a drug or something that's extremely safe, then maybe we can give something that will protect it and prevent people from getting it. So the other question was, is there a test? So the answer is there's no blood test for it. There is a scan called a DAT scan, which can look into your brain and like the PET scan and tell you how much of your dopaminergic neurons are, how they're doing. And if they're down, if your dopamine neurons are a little bit less, it means that you either have Parkinson's or one of the diseases like Parkinson's, but it can't separate it. Now, the problem with that, you say, well, why don't we do it in everybody? Well, the way they figured out that test is that they say, well, how do we know who really had Parkinson's? And they ask people like me, the specialist. So the DAT scans turn out not to be any better than a good Parkinson's specialist by definition. So a Parkinson's specialist is going to be right most of the time. Occasionally a DAT scan will help. There's certain patients that we need to use it to help differentiate it. But the truth is it's a clinical diagnosis, except for some unusual cases.
Okay, thank you. And David Soleil has a question. He says he grew up in Oregon and consumed his body weight in apples, but probably bathed in paraquat and the two pesticides, I imagine. Yeah, Meneb. You want to come up? You want to ask your question, David? And have these two pesticides been confirmed to cause PD?
Yeah, is this paraquat was the other one, Maneb?
Maneb.
Yeah.
Mm-hmm.
So somebody's very knowledgeable about this. So the answer is that there's very little doubt that those contribute to the risk of Parkinson's. So those are two of the pesticides that we find increase the risk considerably. It's so convincing, the data is so convincing. We've been working at trying to get it off the market for years now. And for a variety of reasons, I probably don't have to explain to people, the EPA is not very active these days in getting rid of toxins and regulating things. So there's actually a law firm out there that's an environmental law firm that helped sue the tobacco industry. And that they were one of the people that helped minimize exposure to tobacco. So they've taken on the company Syngenta that makes paraquat, and they're actually suing them. And the goal of the suit is to get them to take it off the market. So I'm actually been volunteering for 5 years, Dr. Ritz, to try to help, because the only way to get these things off the market is going to be to sue them and make it financially better for them to get it off the market. So these are going to hit the press probably in the next year or so, depending on how things go. I think everybody knows about the Roundup lawsuits for lymphoma. That was actually Dr. Ritz, my collaborator, who is the expert witness that helped get those lawsuits through. So it turns out that, you know, going through the EPA, we've tried to do it regulatory, but it doesn't seem to work. So we volunteer our time to get these people, to try to get some of these nasty chemicals off the market. So the answer to your question is, as I said, I don't think it's the one sole thing caused the Parkinson's, but I do think those likely contributed if you were exposed a lot.
Thank you. Donahue, you're off mute. Did you want to, you had a comment you wanted to make?
Yeah, definitely. Hi, Jeff. My name's Aaron. I'm the new director at the Collins and Katz YMCA down the street. Thank you so much for what you're doing. We actually have a few members of our Parkinson's, our Plus program class on the call today. So hopefully you've given them some things to think about.
It's a wonderful thing for so many years, and Anne and so many Rotary people. There's I don't know, Wally Fishman and Henry Sang and all these people, wonderful friends.
Yeah,
thank you for continuing.
Yeah, thank you. Your comment about physical activity definitely resonates. And Rachel McCoy Bedford's on the call with us today. Rachel, did you want to say a few words about what we're currently doing with folks at the Y, and maybe have a question for Dr. Bronstein?
Hi, Jeff.
Hey, Rachel. So we're two of the founders here, huh?
Yep, we go back to 1996. Was when I first met you, when you had black hair.
Yeah, your hair hasn't changed any, even in your lock-in. I'm impressed.
Yeah, our program is thriving. In fact, I got some of the members today to chime in on the meeting because they really wanted to carry on doing a little exercise. And so I am just meeting with still some of those people in their homes, just friends, just doing some exercises.
Wonderful.
Because like you've always said, it's scientifically proven that exercise helps.
It is. There's been, I really feel terrible because I forgot to put these slides in, but there's been a couple of randomized controlled trials. And if I just black out exercise and put in drug X, you know, everybody thinks it would be $100 billion drug because it really slows the disease progression down, and it's really just exercise.
I really notice it when the people are routinely coming to class. They're usually the ones that are being able to move and even cognitive function. We do little brain teasers and memory games, and
you took the words out of my mouth. Physical exercise, actually, the Alzheimer's world has found the same thing for memory and that sort of thing. So somehow physical exercise and mental exercise really help both the motor and cognitive thing. And, you know, I'm not in my lab. This is actually one of those fake backgrounds, but I have a little gym at home, and now that I don't have to drive to work every day, I've really been able to up my exercise considerably. And it's amazing how effective it is in making you feel better. So.
Absolutely.
And thank you. Did you have any other comments, Rachel?
At the very end, I could actually do a couple of exercises and liven people up in their chairs if they don't have to run off. So when you're done with everything, I can actually move people around a little bit, show a few.
That would be wonderful. Thank you so much. Sure. Can I just ask, who is on 310-285-4555? I just didn't. Who's that? Somebody's talking. Okay. Oh, hold on. Who's on?
4805.
4805. Who's on that line? We're just curious. We're speaking up. Okay. Well, anyway, well, I just wanted to thank you so much.
There's one other question I would like to answer. I
Yes.
see here. It's a comment on Norian.
Oh, okay.
Yeah.
Oh, yes, I see it now.
Yeah.
From
I wrote it. I would say that I used it, and I was the walls of breathing in people's faces were melting in 3 days. I had massive hallucinations. The 3 days of being on the door was very, 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 very strong. Yeah. So it's very.
Yeah. So Norians is a new drug. I didn't get a chance to talk about it.
Yeah.
It helps. It helps with the dopamine neurons respond better to the dopamine. Now, it is like all medicines, they have potential risks, and it potentiates the levodopa. So sometimes you actually have to reduce some of the other medicines with it. Some people, it's not perfect either, but it can help people for the, what you call that, wearing off.
Yeah.
So it's just one other drug. Again, it's not perfect for everybody, but it's one other thing to be able to try.
Okay, it comes in 20 and 40 milligrams. I took 20, and 3 days I was just shocked. I was hallucinating so bad. I was really shocked.
So it obviously wasn't a good fit for you, but it's one of the, it's again, there's a lot of different, it's another symptomatic therapy that we're working on.
Thank you very much.
All right.
Well, thank you so much, Dr. Bronstein. Diane, good. I'm unmuting you here. Did you want to say goodbye to Dr. Bronstein? We have a gift for you that we give to our speakers. And Diane, do you want to tell them about that?
Well, you can, Marcia, as well, but it will make a donation in your name to our Polio Plus program from Rotary International.
Thank you so much. I couldn't be the best gift in the world. Thank you so much. It's really a pleasure. And, you know, I've been, you know, involved with your Rotary since 96. So it's been really been wonderful, and I appreciate you know, everybody that's all the things that you guys do in Rotary, especially for our Plus patients and by Carol and all those things. It's just been wonderful collaboration, and we're all growing old together. It's really nice. So.
Okay. Well, thank you so much. I wish you could see us all applauding for you here. It was a great program. And
we can do this in person soon.
Rachel, can you hang on just 1 second? Just Steve Day would like to make an announcement about next week.
Well, I have the privilege next week of presiding. So what I'd like everyone to do is invite their spouses, significant others who are sheltering at home with them to participate on our meeting next week. And it looks like it's going to be a great program. So something just to point out to everyone about Dr. Bronstein, he's actually an honorary member of our club. We made him a Paul Harris Fellow and an honorary member a number of years ago. So it's like one of our old time members is back visiting us today as well, which is great. Thank you, Diane. And Marcia, and we're looking forward to next week's meeting on Zoom.
Okay, well, thank you. Rachel. Yes. Yes. Was somebody about to say something else? Dr. Bronstein, you good? Rachel, would you like to show us some exercises here? You've got the floor. You've got the stage.
Does everybody
I'm going to
see
say
me?
goodbye. Thank you so much.
Oh, bye, Jeff. Till we meet again.
Yeah, take care. Be safe, everyone.
Yeah, send everybody down to the Y. Okay, guys, if you can see me and you're sitting up nice and straight in your chairs, this is something that is probably taken a little downhill, our posture. So take a deep breath. Everybody breathe in. I should be able to see. Oh, there we go. Take a big inhale through your nose and blow out through your mouth. Good. Take a deep breath. That's what we've also got to try to concentrate on as you're sitting around. Nice deep breaths. And reach up to your ceiling and take a reach. And reach high. And there you go. And reach. And reach. Take both those arms up and then over to the side. Hopefully you feel that down under your armpits, down your side, and over to the other side. Good. And up and over. So what's important is trying to fight the rounding of shoulders. So see your shoulders elevate up and down. Shoulders up, shoulders, shoulders. Can you pull your shoulder blades together in your seat? Maybe even grab the back of your chair, which is a really good thing to do if you're stuck at a desk, still managing to do some work. Grab the backs of your chair, bring your chin in, open across your chest, probably feeling that. Good job. How about we sit up straight and you twist and you grab the back of your chair and you can help yourself turn and keep some range of motion in your spine. Maybe a hand on the opposite knee and you're looking behind. Good job, come through the middle. I'm usually used to having some background music. It sounds so quiet right now. I told everybody to be quiet in the house. Okay, so you're gonna twist around the other way. And turn. Good, and then you're back in the middle. How about sitting up straight, drawing in your navel to your spine, sitting up on your haunches so that you have your shoulders over your hips. And then a slight bend, almost like you're gonna pick up something from the floor, but you're not gonna lean forward. So you've got your waist moving a little bit, side to side. There you go. Keeping up nice and straight, keeping your head between your shoulders, guys. There you go. And, all right, in the middle. When you are sitting down a lot, you can't see my feet, but it's really important to pump your blood back up. So you toes up. Like if you were sitting on an airplane for a long time, you pump your toes, you pump your heels up, anything you can with pumping your blood back up to your brain. Otherwise, when you sit a lot, it's all gonna pull down. So your legs, heels up and down. How about you sit up tall in the chair and we lean back and you bring your knees up and your arms and you do a big hooray. Knees up, arms up, hooray. Knees up, arms up, hooray. Knees up, arms up, hooray. Good, pick it up, lift. I could really go on for another whole hour, just like Jeff. We never have enough time. So I'll even put it out there. If anybody is interested in some home exercises, then maybe my email can be shared. Because if you're sitting around, I think just these similar things, I didn't even get to really stretching, but there is so much to do. And it's getting really hot outside, so walking's gonna be a little bit of a problem. So it's really nice chiming in on your Rotary meeting. We go back a long way. Our YMCA and the Plus program, just as Jeff said, 1996. So some of you out there, like Ron, we all like get together now and again. So I'm the exercise lady, so I'm willing to share if anybody needs help. And when we do get back into somewhat of a normal life, remember our YMCA, because that's where we hold the program 3 times a week. And it's a marvelous facility. So if you are in the around West LA, come and give us, come and look in. Good to see you, Erin. All right, I'll say goodbye to everybody. Marcia?
Rachel, so we can give your email out to everybody and you.
Yeah, I'm gonna just say it. We don't know how long we're gonna be cooped up. I put it out there. And it's, you know, a lot of people are Zooming now. A lot of regular gyms and health clubs are offering Zoom exercise. So, yeah, I've been doing 3 times a week at 12:30. We're just chiming in on some exercises. It's
well,
nothing.
Send you an email. I'd like to get some of these exercises.
So, yeah, you can give my email out if anybody emails you and they want to know.
Okay,
sure.
Thank you so much.
Okay.
All right. Well, everybody, I'll just ding my head here. Ding. We're gonna be we're gonna end the meeting. Ran quite long, but it was a great meeting with so much information. And thank you, Rachel, for hanging around and all of this. And we'll see you at the YMCA. And until next time, I'll see you guys later. I'll see you next week.
I ask you one last question?
Yes, one last question.
For a while,
it's the Rotary Club. She's the Rotary.
No, I want to talk about
what? Boxing.
Gosh, right. Boxing became the real thing, the rage, I would say, over the last couple of years, maybe more. So, of course, anytime I see the word Parkinson and match it up with something that seems to be working, I'll do it. So I've been taking, I can't forget. I've been taking boxing probably for what, 2 years, Rachel?
Yes.
About 2 years. It's been amazing. It's been amazing. And it's hard, and I'm really boxing.
Are you kickboxing or regular?
No,
boxing?
I'm not. I'm boxing.
Boxing
in the ring.
So, you know, I was just going to say the boxing thing came in. Remember, we've been around doing regular exercise, and in my class, it's everything. We talked about cognitive functioning. So in my all-round exercise class, it's everything. It's voice, it's breathing, it's hands and fingers and posture, and just small motor skills, big motor skills, light weights, floor stretching, wall exercises. So boxing's great, whatever you will do. I'm a certified cardio kickboxer. So the thing with the arms going forward, I'm always trying to get the posture with the opening. So I would say if you've got boxing forward, then you need to do exactly what I showed you in things like the opening of your chest. Because a boxer's posture is going to get a bit hunchy and round because everything is forward. Our whole posture throughout the day is front. Everything is going forward. Our arms are in front. We sit down. Our bodies are closed to the front and they do not open enough. So that's my only thing with boxing. Mix it up a bit. Rowing, other things, a lot of stretching up the wall, doorway stretches, so that you are not encouraging a rounding posture. That's the only thing with that. But I know it's terribly empowering to hit things. Oh,
I have
great.
a pink and white one. I got pink and white.
Anything, moving is anything that you can do. What you like, you do.
Fun, right?
Good job, everybody. I hope everybody's moving.
Wow, you're terrific.
Does anybody else want an exercise question while I'm here?
I know, I'm going to send you an email, and so I can get onto your other, see what you're doing, you know, so that I can't come to the YMCA or anything right now. But I'd like to do your, 33 times a week or something?
I'm doing Monday, Wednesday, Friday, 12:30. We were limiting it to 40 minutes. You know how Zoom goes. They're requesting, I'm going to pay that fee. I'm going to do the Zoom, and we're going to go a little longer than 40 minutes. By the time we all get on, I've spent an hour with people on the phone just trying to help them set up with Zoom. So for some people, the new technical stuff that we have to now do in our own homes has been a little bit of a problem, but hey, I'm at home, and you know, I'm doing what I meant to do, and I'm actually exercising more than I probably did before, but then I'm eating more too. So
yeah. Okay, well, maybe you should come back and be our guest speaker. You know?
Hey, I could fill up a few hours, just like Jeff. We're good talkers. We're talkers.
All right. Well, thank you, one and all, and we'll see you next time. Rachel, come to our meetings anytime. Okay.
Sure, I'm sure. Yeah, Erin will drag me down there, I'm sure.
Talk to you later.
Marcia?
Bye.
Marcia, remember Steve Day said, or somebody said we need a speaker in June. Put the exercise.
Speaker in June. Rachel. Yeah. We'll talk to Steve Day. We'll
okay, if we're allowed out. Okay.
Talk to you later.
Thinking.
Yeah.
Or, or in home, yeah.
See you guys all later.
Okay, bye guys.
Bye-bye. Gosh, look how many people are still here. Do you guys want to? I have to go to a board meeting now, so I have to sign out of this meeting to get to the board meeting, okay? So I will talk to Peter. Great to see you. Yeah.
Marcia, are we having a board meeting right now?
A board meeting, but
on the same
a different address. I think. Well, Diane posted.
Oh, it's different.
Yeah, it's. I've got it right here. Zoom. Go to 977-583-25794.
25794.
25794. 977.
All right.
583-25794.
Is there a password?
She didn't give me one.
Okay.
I just have that. All right.
Thanks, Mark.
Okay, bye. See you in a minute.